Yeah, yeah, it didn't really feel safe. So it was
That is
weird.
normal for me. I don't know, I'm live on YouTube and will not say
Not yet,
nothing.
not yet. Oh, I love it.
I want to say it's normal.
Yeah,
Your feeling, I understand.
yeah, I heard that from other people when we came back.
Well, put it down as an experience.
Yeah, I guess.
Good morning, Mary.
Good morning, Steph, and good morning to all.
Hi.
Hi.
Great. Well, it's 9:32, so let's make a start. Welcome to Neuroccino on this Monday morning, bright and early, 9:30. The rules of the game are that anyone is welcome to bring a paper or a book they got excited about and briefly present it to the best of their knowledge or to the best of their preparation, as we have today. And then we discuss it as a group, talk about it, and see where it takes us. We sometimes end up in interesting places that no one could anticipate. Good morning, Victor. So for those of you who do have a paper, if you could just share it in the chat, just copying mine. And then we can share the screen and talk about it. Sorry, where's the actual paper? It's fine. I'll send you the link as I go along. And that's basically it. Those are the rules. So feel free to come and join us every Monday morning and just do bring a paper along and we can discuss it. I'm just looking for the link now. Here we go. It is this one. Sorry, hosting and presenting brings me to the edge of my multitasking capabilities. There we go, you get the link now. So that's the paper we're going to talk about today, Advanced Imaging and Labeling Methods to Decipher Brain Cell Organization and Function, published in Nature Neuroscience Reviews, I think. Yes, Nature Neuroscience Reviews. I'm just going to share my screen with you so you can actually see the paper we're talking about. Tack, tack. Here we go. Oops, he disappeared on me. Okay, can you all see the screen okay? Lovely. So as I mentioned earlier, it's not necessarily presenting to the best of your knowledge, but also presenting to the best of your preparation time. And for this one, I had minimal time this morning, so I do apologize for rushing it. But the half paper that I read was very exciting already. So it's all about reviewing how we use certain imaging and labeling methods and what the recent advances were that help us to decipher brain cell organization and function. And I actually did learn quite a lot this morning reading through it. But basically there is different ways that the field has advanced, and one is that the technological, methodological side of the techniques has improved. So we're now actually talking about super resolution, which is quite exciting. I'm going to show you an example in a minute. But also the way we can label the tissue and the different levels of the complexity of the brain has vastly improved. So I'm going to jump right to the figure, which I thought was absolutely stunning. Here we go. So here you have an example of the different levels of complexity. We can look at the full brain. We can look at different networks in the brain. We can look at the synaptic level and even dive into the synaptic complex in more detail by looking at receptors and neurotransmitters. And there's various different methods here. As you can see, the techniques here are plotted for their spatial resolution. that's the scale on top, and in blue. And the time resolution down here from hours to milliseconds. And those are the different microscopy techniques available. And you can see that the different techniques have a different sensitivity to time and spatial resolution. And then as an example of what we can get from it, you can see here the whole brain imaging. That's a clarified mouse brain, I would assume, where you can see the entire brain. You can then also look at individual networks in that brain at 200 micrometers. And we can go down to the dendritic spine level. Now we're looking at 10 micrometers. And then even here, a beautiful image that I have to admit is out of my comfort zone in terms of what we're imaging, because we just do not get this amazing resolution with our machines at the moment. But this is a multimodal super resolution microscopy of the synaptic receptor trajectory where several methods were overlaid. So this to me is just a stunning figure that summarizes nicely what we can and cannot do with the different methods, and the level of detail that we can then get for our imaging. And what you can see here already is the combination of the best of both developments, the one that we now have a absolutely stunning resolution, so we can really look at the granularity of the brain at this point, but also the labeling has vastly improved, so you can see the structures at a finer detail. So a very interesting read that I recommend to read the full paper, which I'm definitely going to do later on. But absolutely stunning what is possible nowadays, and how we got there. So it's nicely, it's very well written as well. So it's nicely explained where we started and what has changed and why it has changed and where it brought us now. So it's a very, very nice educational read, I have to say. That's about it. Very brief paper today. And we just discover it as we go along, I guess, together. So do you have any questions at this point?
Yeah, I just wanted to ask.
Hi. I read that there is a paragraph genetically modifying proteins.
Yes.
And we are able with this super resolution also to see visually when the folding, for example, of a protein is modified and maybe related to a genetic sequencing. Maybe we are at that level, I was asking.
Yes, so I was actually thinking of you while I was reading it, because there's quite a lot of hints to genetic methods that you can also use and look at the folding. So definitely don't pin me down on the details for this, but definitely something that you currently want to look at in terms of the genetic factors that you can image as well.
Yeah. Here there is like a box, the box 2, where they say that you can spatially, temporally labeling seems this processing in really the so chemical biological level.
Yeah. So what I thought was quite interesting in box 2 in particular as well was that in terms of using tracers to label cells and what is connected to what, you usually inject a tracer at one part of the brain and then you let it travel along the axonal structure and you see where it goes, so you know what the network should be. But they actually say here that there is a spatial resolution problem and that new vectors can actually overcome this. So it's usually based on a virus that they inject with the labeling tag on it. So yeah, quite interesting, I think, but definitely out of my comfort zone, I have to admit. Any other questions, comments, ideas?
I think we have a question on the chat.
I just saw that. Quick question. They are all structural imaging techniques, right? No, they also talk about functional imaging, but it's not functional imaging in the way that MRI people would understand functional imaging, that you do a task and you get a part of the brain that then lights up due to the task that you performed that requires more oxygen and more blood flow. And obviously the images that we can see here are static images, which imply structure. But because you can look at the receptors as well, for example, and the neurotransmitters that get released at certain times, you can also infer functional imaging, just a different kind of looking at function than we're used to.
I'm just browsing through the article, and I'm so excited by the figures about how the microscopes work and everything. For me, it's always incredible when you see to what length people will go to improve the imaging by just completely changing everything about their materials and stuff like that. It's incredible.
It's a beautiful paper. It's well written, and also the images I thought were absolutely stunning. But yeah, I can see you appreciate the technical side of it as well.
Yeah.
But it's also Nature Neuroscience Review. So I guess at that point, you need to make a good effort with your figures.
I was looking to figure 3, and where you have all the protein labeling strategies. And I was wondering, also in neuroimaging, we always have the question, which is the gold standard. And also, I think that also here, then when you see all these techniques, it's super resolution, but there is also a lot of diversity. And so maybe you see which is the golden one, which is the best one.
So it's quite nice because they do address that at multiple points in the paper and say this is what is commonly done and was considered the best method, but this was the limitation of the method up until now. And then they introduce the new developments and say how they can fill that gap or push that boundary a bit further that will allow us to just go one step further and actually look at something in more detail or something new, for example, as well. So some particles and things can be discovered with new methods that we didn't really look at before, for example. So I guess it's similar to what we're doing with MRI, where what we thought was the gold standard has changed and evolved over time. And the more you combine the methods, the more you look at it, the more you realize where the limitations are. And then you try to find a complementary method to push that boundary a bit further. And that kind of, it doesn't take the gold standard away, but it puts it into a better perspective of what we can and cannot do with it. And from what I read so far from the paper, it's very similar in microscopy, where they used to have a gold standard, but they were quite aware of the limitations of the method. And now they're pushing the boundaries to have less limitations or complement current methods.
On that point, by the way, while broadening the paper, have you seen any method that could be used concurrently with MRI or something like that? Because, well, MRI, I guess, in mouse and rats, but still.
Well, there has been attempts, as far as I know already, where they try to combine microscopy and MRI, in particular for, let me go back to that beautiful figure one here. So for the clarified brains, for example, you get those beautiful reconstructions of the connectional anatomy of the brain because you clear out all the other fatty proteins. So you're left with the connections and the networks. And that has been tried to be matched by MRI, but obviously the resolution at the moment was not at at the same level. But there's a big push because the resolution of MRI is greatly improving, especially when we look at like animal scanners rather than human scanners, and post-mortem rather than living. We had many papers on that discussion at Neuroccino. So there is definitely efforts to try and combine it. And then obviously all the work by Carl Zillis and Katrin Ammon's group about the receptor mapping in the brain. So the different fears, and especially the wet lab and the dry labs, are kind of coming together in the near future, and there's a big push to try and make it happen. But at the moment, I think this is actually quite telling, because we have exactly the same figure for our brain imaging methods, where we can map the spatial and the time resolution, and we kind of have to bring that together to overlap in space or overlap in time in terms of the resolution to combine it properly. And there's a big push to go in that direction.
Yes, I guess that also there is a big push in the field of diffusion imaging, when you try to try to estimate the microstructure, the fine microstructure in an area based on the signal you get from diffusion and all the diffusion techniques. But yeah, I don't know if we are that close to it, but I know that there is a push, and it's I hope that we will find something interesting, very interesting soon, because that could really revolutionize everything in terms of human research. Because although we can here like try and test methods and like MRI methods and see what's inside the brain exactly immediately after, if you want, because we are working with animals. In human, it's much more difficult. So I don't know if there is many people who did a brain scan just before passing and have their brain extracted. So it must be like very small sample, and yeah, it's hard to test the hypothesis with this kind of data. But with this, if we can really link the two together, it's exciting.
It is absolutely exciting, yes. There's more and more data available from people who were imaged before and after. But obviously, yes, it does come with its own limitations. But also, maybe we don't need to go from diffusion to synaptic receptor complexity in one go. We might just go and take one step in the middle, which, for example, with polarized light imaging and those kind of methods, we might just take it step by step and make it happen to maybe meet in the middle, for example. Chris.
Yeah, so in MRI, super resolution is already used, but instead of using completely different methods, they use different modalities to improve the resolution of the images, especially, for example, when we are missing a lot of slices, or where in clinical neuroimaging, you don't necessarily need all the slices in one axis. So you will only have like 20 voxels, or sometimes even less, in one axis. And with the super resolution and another modality, you can fill up the missing slices. So then you can have a much better image it's a reconstructed image. Then you can use it for more accurate analysis after that.
Yes, absolutely. But like the end game would be to map the entire brain at a super resolution.
Yeah, but
and also combine.
Well, I mean, super resolution can also help to reconstruct pretty bad images. I mean, clinical images are pretty rough sometimes. So that's also a very, very exciting output of the super resolution algorithms, because it can really fix some pretty bad images. I mean, it's not magical, but like you can use these images almost at a research level, like a research level imaging.
Okay, that's what I wanted to ask, because you are basically modeling based, we've got missing data and you're modeling based on other data, what should be there. How sure are you that you're actually modeling what is there and not what is generally there when you think about it and you look at the different things? It's different from, because as you said, it's not magic. You cannot exactly know what it is there definitely. So how close it is.
Well, I haven't. Well, I used the super resolution, but I didn't really get into the details of the methods.
Okay, all right.
But
I guess that brings us back to what is the gold standard? How do we know what is there, what should be there, and what is missing in a clinical data set, for example? But also, like clinical data, I think is always a special case. But also, before we go into clinical data set, we should know what the brain looks like when it's healthy and well functioning. And also, by combining the different levels of granularity in the complexity of the brain and different methods, we can actually combine like cortical areas, receptors, connections between areas, and actually get a whole brain, whole system understanding of how the structure and the function work, which will still take us a couple of years. Otherwise, we will be out of a job tomorrow.
Yeah.
And then bringing it to the clinic, obviously, and I'm not saying like we have to solve one first and then move on. Is definitely worth doing it in parallel and benefiting back and forth. But yeah, there are still many gaps that we need to close in our understanding of how it works and the methods that we can use for it.
Also, that's something about the clinic perspective.
Mm-hmm.
I always wonder what makes the image that we usually get so poor, because I've seen a lot of clinic images, and it's not only because of the pathology that the image are poor. Sometimes just the scan is bad, and I was wondering, because I'm not from this world at all, so I'm honestly wondering, why is that? Do you have any insight about that?
There's it's not just one reason. There's
right.
many reasons. Starting with the equipment in the hospital is usually not as advanced as the scanners that we use in research. Then another reason is that the interest in most cases why you do acquire the clinical scan is not a research question, it's a clinical question. And in order, for example, if we take stroke, the thing you want to know in the first instance is there a bleed or is there an occlusion? For that, you don't need a fancy super resolution scan. You can take a CT scan just to see if there's a bleed. And then you plan your treatment pathway accordingly. Now, obviously, for our purposes, those images are not great, but they completely serve the clinical need at the time. Another reason is timing. So usually we have the luxury in most institutes that we have scanner time available to us, but in the clinic, the scanner is used all the time. You don't get easy access to it. So you don't have an hour to scan your participant. You have like 5-10 minutes. So obviously the acquisitions you can get in that time do not compare to research acquisitions. And then on top of that, it's patients, it's people, don't forget. So especially in the acute stages of a disease, people are scared. People don't want to lie still. Like, it's very hard, and it's a strange environment, especially if you've never had a scan before, and then you have your first scan in an emergency setting. You're not just going to lie there and say, oh yeah, just fire away, take as many images as you want. I'm happy to lie here for an hour not knowing what's going on with me. So there's a lot of movement and anxiety and all these factors surrounding taking a scan. And if you put that all together, you get an image that has suboptimal quality in the end.
Okay, thank you very much. That was very insightful.
I don't know, Leah, do you have something to add to it?
Yeah, no, what you said is really true. Like sometimes when you are in a clinic and you have to just do a sequence in a clinical settings, they say, yeah, we don't have time. Like we have other priorities in that moment. And so you have to step back. And also, usually, I think there is also maybe a difference in the training. So maybe for example, also a simple DTI measures as FA and MD, now they are entering in the clinics, but a lot of people also that are trained in imaging, they don't really are trained to use those measures. So or also fMRI, they understand fMRI. It's intuitive because you see activations, but when you came to parameters, measures, there is a lack of training. I think we should also train. If we want to move in a super resolution, we also need to do specific training to both the medical staff, also the people who are acquiring the images. Usually, in my case, I'm a physicist, so for me, if you change parameters, is kind of important because you are trying to fix all the measures, and sometimes there is a person with, I don't know, a big nose, or an artifact, and there is the person who move all the field of view and change parameters, and then for you that patient is not useful for your study because everything is changed. But for them, it's the best imaging that they can have in that moment. So it's difficult to argue when then you have just 5 minutes and you have to go. And so, yeah, a lot of things going on. I mean, that it's really stressful morning if you have to do acquisitions.
Another factor is also that we, and that includes everyone currently in this live meeting, we spent an awful lot of time on the post-processing and processing of our data. Well, from our perspective, it's a pre-processing, but from an imaging perspective, it's after you acquire the scan. So we do spend a lot of time preparing our images, correcting them for motion and for other distortions. And in the clinic, usually you acquire the scan and you push a button, and the software that happens to be on the scanner pre-installed just does its black box magic and gives you an output. And you're not as much in control in terms of how you can influence the processing and the parameters, as Leah said, that you can also change in the processing of the data. And that usually is a very quick process. I think it takes less than half an hour for like tractography, which is something that wouldn't happen when we process the data, for example. It takes us a much longer time. But again, it's the clinical need, right? It's what they need, and they need it fast. They don't have time to just faff around and say, oh, let me do top up and eddy correct and all of the different things now, Gibbs ringing and all of those. It just takes too long. And it also, depending on the question you have, obviously, it doesn't serve the clinical question because you're not interested in, oh, is this exactly the right location, or does this track go there or there? You just need to know, is it still there? Is it damaged by the lesion, or is it fine? So the needs are very different, and therefore the end result is different.
All right. I also assume that there are norms, very specific norms, that what you can use in the clinical setting, because it's linked to health and everything. And so you have to validate it and everything. So you cannot really use newish techniques and sequences. So you can't really explore this kind of thing. And yeah, all right.
It depends. So to the best of my knowledge, for tractography, for example, or diffusion imaging, the only algorithm that is approved in the clinic is the FACT algorithm. That is now 10, 15 years old. at least. And we, like in terms of research, we moved on from that quite a while ago, but in the clinic it's still a commonly used algorithm. And it's often okay because the data that is acquired doesn't have 100, whatever many directions. They have 6 directions or 25 directions, right? It serves the data better than what we're doing. But usually, like the rule of thumb is it takes about 10 years for something from the research side to be translated into the clinic. At least 10 years, if not longer. You can see that. You can definitely see that. But yeah, it does take time, and if it doesn't serve the clinical purpose, it takes even longer. That said, the limitations we just mentioned mainly apply in the acute setting. If you image clinical patients in the chronic stages, you have usually more time because you pre-plan the scan. It's a lot less of a rush. Sorry, our little mascot's making a mess behind me.
Yeah, I was thinking the same thing. Then if we have, like, for example, neurodegenerative patients that are, you monitor them in times during, so you prepared, as you were saying, the scanning, so there you have more time, they are more used to the scanning, you can do also some, you can enter in like an imaging study, something like that, or also before surgery. Usually they really prefer to take the patients to have the better imaging as possible, because then you have to do the operation. So in that case you can really spend up to 2 hours for a patient, because it's the most important thing that you can do before the surgery. And so it really depends on the pathology. And also I think that now with the new scanners, we did a big jump forward, because now you can have really 60, with also parallel imaging, acceleration factors, a lot of for diffusion, a lot of directions now are feasible in the clinical routine. Also for when I started and now I saw like a big jump forward. Now we are more close to the research resolution, I would say.
Mm-hmm. What's also great is that clinicians, at least the clinicians I came across, and there might be a slight bias because I always worked in university hospitals, but those clinicians are very interested in new development in terms of techniques and methods that they can potentially bring back to the clinic, and they try to push those boundaries, or try and allocate more time to get better imaging. So it's very important to have that multidisciplinary integration between the theoretical research side and the translational clinical side, which, depending on the centers, it can be so-so. But my experience has been that clinicians, especially neurosurgeons, are extremely motivated to try and get the best possible imaging for their patients, and implement it in their care where possible. That's a much longer answer than you expected, Victor, isn't it?
Actually, I'm thrilled because it's really not where I came from, because I mostly work with healthy participants. And I very recently had an experience, well, with you in the when we had to draw the lesions directly onto the scan, and seeing the mess that it was, and yet it was still, I think, quite a nice data set.
Uh-huh.
So I imagine it can be much worse, and I was surprised, honestly. And now, I got a much better understanding of what's going on, and thanks for that.
So you were lucky, because the data that you got to work with was very clean, tidied up data set already. When you start from scratch and you have CTs and MRIs of different resolution. That's what I'm
also because there are all these factors in the acquisition that can make a scan go badly. When you're a researcher and it's like your data set for your PhD or wherever, you have to go always in person to check every step. You have to arrive before in the scanner. You have to follow every step since the image is saved and you get it. Otherwise, it's a lot of work. I think it's. You really need a lot of people working on that. I think that we should make maybe a knowledge that the research need staff, really prepare staff, because it's not easy. And sometimes it's not really understood.
I can second that. So for my PhD work, I was sitting there for every single scan, just writing down the order of sequences. So you don't just run an automatic thing. Well, it's also a while ago, so running automatic things wasn't really that reliable at the time. It's much better now, but you really had to go and sit with the radiographers and observe the scan to make sure that everything is done by the book and always the same, and to know what's going on, what's happening, and also pick up on problems during the scan quite quickly. For example, because I was doing acute stroke, we had one patient who the heart rate, so you monitor the heart rate, and the heart rate went through the roof. So I was like, we got to stop the scan, we got to get in there, something's wrong. So we stopped the scan, rushed into the room. Turned out that patient just had to cough, and because we told the patient not to move, they were trying to suppress it so badly. The heart rate went up. So it's always good to be there, have a second pair of eyes, and an extra brain to just help monitor and react to situations. But obviously that's the acute stage. Chronic patients are much easier to scan. It's more similar to healthy controls, depending on the pathology, but it's way easier. You take that urgency and stress away if you don't do acute. On both sides, the patients and the research side. But ideally, and this is why I thought this paper was quite exciting as well, ideally, one day you come into the clinic and you get a super duper scan that takes 5 minutes and gives you all the answers in terms of structure, function, and different levels.
Yeah.
Sorry, no, let me take that back. Ideally, you don't even have to come to the clinic. The scanner just comes to you.
Oh yeah, that's much better.
Which is where we're already going. There's already portable helmet-only MRI scans.
Yeah, it's what, 1 Tesla even, not even, I don't know if it exists. Yeah.
I don't know. I know we bought one in my old institute a while ago, but I don't know the specifics about it. But like, again, I'm completely biased with my stroke background, but time is brain. So if you can actually have a helmet scan in the ambulance, for example, you can already acquire a quick image to see if there's a bleed or an occlusion. So by the time you get to hospital, you already know what the treatment plan is, and you don't waste time on hoping that the scanner is available by the time you arrive and taking the images. That all takes time that you could already preempt during the drive to the hospital. So yeah, obviously with time, I think that spatial and timing resolution will see massive improvements. But then obviously getting to that level where you can have a quick scan of synapses and receptors, that's a long way. But multimodal integration is also a big field where we're trying to go and try to bring it all together rather than just have a super resolution but not integrate it with the function or the tractography, for example. So it's all coming together, I think, and there will be very exciting developments in the years to come. It's my hope and my understanding, but we'll see. And depending on where your trajectories go, you can be part of this excitement.
That might be nice, yeah.
I see the excitement and the fear equally in your faces.
No, I was thinking that it's so beautiful what we do in research, but for a clinical translation, we need information and investment, because otherwise it's just an elite. It's really important to inform people about these methods.
Which is what we're doing, and that's why university hospitals are a great starting point, because you have that close collaboration between the clinic and the research side.
Yeah, that's the way.
And all it takes are people who are curious on either side. So we need clinicians who are interested in new developments from the research side, but we also need researchers who are interested in making their methods have an impact in the clinic. And if you have those two work together, I think we can go a very, very long way. Scanning while driving has a problem of motion artifact. Fair point. Fair point. I was talking about an ideal world. Maybe there's like a suspension thing you can do. I don't know.
If you have a uniform motion and not acceleration, for the relativistic point of view, you can do that. But you need no acceleration.
Depends on the driver.
Yeah.
Just screw the skull to the machine. Bit invasive. Bit invasive. I'm not sure it's going to flow. But that's the way to go, people. Just think, be creative. Just please don't screw anything in the skull. Especially not as a physicist, Victor. Bad idea. Bad idea.
Yeah, actually, they do that for some, I think it's not for scans, it's for some surgeries and stuff like that. They have to screw the skull, right?
For some of them, yes, but it's usually neurosurgeons, not physicists. You do not get to do that. Unless you take another 15 years and retrain, then we can talk about it. Leah, we need to make sure that we take all the toolboxes away from the office. Victor's on a mission here.
What?
We need to remove all the toolboxes in the office.
Okay.
No more screws and screwdrivers around Victor. Right. Any other questions, comments about this paper? No? Okay. As I said, I'm sorry I couldn't prepare it in more depth, but it's a beautiful review, so do go and have a read. It's well written. The images are stunning. So it's not a difficult read, which we also sometimes have here. And it's definitely very exciting science, and will give you hopefully good ideas of where to go in the future and how to change the way we do our imaging. Right, with that, let me just say it's the first Neuroccino for July. We have a very exciting month. There's new videos already on the channel. There's Meet the Editor with Brain Structure and Function. It's an interview with Laszlo and Michelle, so the outgoing and the incoming editor-in-chief. There will be a special Neuroccino later in July. Again, Meet the Editor for Nature's Communications Biology. So that's going to be exciting, where you actually get to ask your questions and interact with the editor yourself. Then we have a CNS talk later this month. And I think that's it for specials coming your way in July. You should have all received the newsletter. So you have the titles, the links, and the dates in there as well. And obviously we will advertise them closer to the time as well online. With that, thank you so much for coming today and for the wonderful discussion. I very much enjoyed it. And I wish you an exciting week in science, and I see you next Monday. Bye.
Bye.